IB Bio, Transcription Translation Part 5. We'll review translation with just a bit of added detail. Once we review translation, we will examine the nature of mutations, specifically the sickle cell mutation. The essential idea is that information transferred from DNA to mRNA is translated into an amino acid sequence. Let's start with 3 important IB syllabus statements. Explain the process of translation leading to polypeptide formation. Explain the process of translation including ribosomes, polysomes, the start codon, and stop codons. Remember that peptidyl transferase is the enzyme that forms the covalent bond called a peptide bond between amino acids at the ribosome. Lastly, state that translation occurs in a 5-to-3 direction along the mRNA. In this diagram of translation, you can see the 3 tRNA binding sites on the ribosome, the A site, the P site, and the E site. An empty tRNA molecule departs the E site and moves off into the cytoplasm. An arriving tRNA molecule attached to the amino acid glycine enters the A site. The anticodon of the tRNA molecule is complementary to the mRNA codon seen here. In site P is the tRNA attached to the growing polypeptide. Once the tRNA with the glycine enters the A site or binds to the A site, peptidyl transferase will form a peptide bond between glycine and phenylalanine. The polypeptide chain will transfer to the tRNA in the A site for a brief moment as the ribosome translocates left to right, opening the A site for the next arriving tRNA. Now, in the images to follow, the ribosome will not show 3 binding sites, the A, P, and E. Keep this in mind. You need to be aware of 3 sites, and the E site is often neglected in diagrams of translation. Translation starts with the AUG start codon. Translation stops when the ribosome reaches a stop codon, such as UGA or UAG or UAA. We can see the P site with its tRNA holding the polypeptide chain. A newly arriving tRNA sits in the A site, and you can see a peptide bond forming between the amino acids. The enzyme within the ribosome, known as peptidyl transferase, forms that peptide bond, and for a brief moment, the polypeptide chain is attached to the tRNA in the A site, but then the ribosome shifts to the right. Here we see 2 ribosomes on the mRNA. This is known as a polysome. 11 codons are identified. This would mean 33 RNA nucleotides in 11 triplets. We can see the amino acids in order according to the mRNA base sequence. We can see peptide bonds forming and an arriving tRNA with amino acid 8 soon to be added to this polypeptide chain as the ribosome translocates to the right. I like this image of mRNA and the tRNA molecules because it clarifies the relationship of the codon to the anticodon in terms of purines and pyrimidines, pyrimidines, and pyrimidine-purine. You can see the formation of the peptide bond by the enzyme peptidyl transferase. Now I know this is the 5 end and this is the 3 end, and how do I know this? And how do I know the ribosome would be moving from left to right? Because the A site is here as the tRNA holding the polypeptide chain is in the P site. Arriving tRNA molecules enter the A site, thus the ribosome must be moving left to right. This is a very straightforward image showing clearly the complementary relationship between the codon and the anticodon. When a codon shifts into position A at site A, the codon determines which tRNA and thus which amino acid will come into place. The fidelity in translation, the synthesis of an amino acid sequence according to the mRNA nucleotide code, is maintained by the complementary codon-anticodon relationship. In this micrograph, we see polysomes, one here and one here, an mRNA strand with numerous ribosomes, each of which is translating the codons into polypeptides, which are visible here. Here's a new IB syllabus statement. State that translation consists of initiation, elongation, and termination. It's not complicated, but let me explain using images for initiation, elongation, and termination. Initiation starts with the codon AUG, and the tRNA attached to methionine joins the mRNA even before the subunits of the ribosome have joined. In this case, the mRNA is bound to the smaller subunit of the ribosome, and the larger subunit has yet to join the complex. Then, the large ribosomal subunit joins the small subunit. The tRNA attached to the methionine amino acid sits in the P site. The A site is empty and ready for the next tRNA. Translation is an energetically expensive anabolic process. A lot of energy is required in the form of GTP and ATP. Now, we don't need to focus on the extent of the energy requirements, but keep in mind that translation is the synthesis of a polypeptide. Energy is required. Here is another image of initiation showing the start codon, and over here to the right, a stop codon. The tRNA carrying methionine is attached to the mRNA along with the small ribosomal subunit. The large ribosomal subunit has yet to arrive. Now, elongation is simply the process whereby the polypeptide chain gets longer. You know this well already. A tRNA arrives to the open A site. A peptide bond is formed between the amino acids. The polypeptide formed is then attached very briefly to the tRNA in the A site, and then the ribosome translocates left to right in this example. Thus, the A site becomes open, ready for the next tRNA, and an empty tRNA exits the E site. Here is another image showing elongation. A site is open, a tRNA arrives, peptide bond is formed between amino acids, the polypeptide attached briefly to the tRNA in the A site. As the ribosome translocates, the tRNA in the A site shifts to the P site, the A site's now open, and an empty tRNA molecule exits the E site. Termination. The ribosome reaches a stop codon for which there is no complementary tRNA. Proteins, known as release factors, play a role in ending translation. The components fall apart, polypeptide synthesis is complete. And that moves us to an IB syllabus statement that we studied a long time ago. Draw and label a diagram showing the structure of a peptide bond between two amino acids. Here is a diagram showing the formation of a peptide bond between two amino acids. This is condensation synthesis. The bond forms between the carboxyl carbon of this amino acid and the nitrogen of the amino group of this amino acid. Here is a dipeptide, and you can see the position of the peptide bond between one amino acid with its R group here and the other amino acid with its R group here. In this diagram of translation, the amino acids are shown in very high detail, and each is identified by its R group, as you can see here. You can see the formation of the peptide bond between the carbon of this amino acid and the nitrogen of this amino acid. Here's another diagram of translation showing the specificity of the peptide bond formation between the carboxyl group of this amino acid and the nitrogen of the amino group of this amino acid. This tRNA cannot yet arrive to the A site because the ribosome has not yet translocated in the 5 to 3 direction. Here are two IB syllabus statements that should refresh your memory from our earlier study of proteins. Describe the 4 levels of protein structure. Explain how secondary and tertiary structures are stabilized. Here is a polypeptide consisting of 4 amino acids held together by 3 peptide bonds. The primary structure of a protein is held together by peptide bonds. The primary structure is the number and order or sequence of the amino acids in a protein. Here's an image of a polypeptide with 5 amino acids in a sequence. You can see the R groups very clearly here. The primary structure of a polypeptide is the sequence and number of the amino acids in the polypeptide. The secondary structure of polypeptides includes the alpha helix and beta pleated sheets. Both of those structures, the alpha helix and the beta pleated sheets, are stabilized by hydrogen bonds. The alpha helix of a polypeptide is secondary structure and is structurally stabilized by hydrogen bonds. The tertiary structure of a polypeptide would be described as the folding of the secondary structure. You can see the pleated sheets here with many folds and the alpha helix here with folds. The complex shape of tertiary structure is stabilized by the interactions between R groups. These 4 forces maintain the tertiary shape of a polypeptide, and those forces are hydrogen bonds, ionic attractions, nonpolar interactions, and disulfide bridges. In this image, you can see the primary structure, which is the number and sequence of amino acids in a polypeptide. We can see secondary structure that includes the alpha helix and the beta pleated sheets, both maintained or stabilized by hydrogen bonds. The tertiary structure is the folding of the alpha helix or beta sheets, and you can see that here, as the polypeptide folds back on itself with the alpha helix and the beta pleated sheets, and the tertiary structure is stabilized by hydrogen bonds, ionic attractions, nonpolar interactions, and disulfide bridges. Now, the quaternary structure, or 4th level structure, is a protein composed of more than one polypeptide chain. Here's a nice image that displays the 4 forces that stabilize the tertiary structure of a polypeptide. We have hydrogen bonds, we have ionic attractions, we have non-polar interactions, and disulfide bridges. R group interaction stabilizes the 3rd and 4th level of protein structure. Here's an IB syllabus statement that extends your understanding of 4th level structure. State that quaternary structure may involve the binding of a prosthetic to form a conjugated protein. Stay tuned. Hemoglobin has quaternary structure with a prosthetic group that's formed a conjugated protein. The prosthetic group is the heme group, of which there are 4, each of which includes an iron atom that binds oxygen. The polypeptide with the prosthetic group forms a conjugated protein. So with this slide, the details of transcription and translation are behind us. You can review both processes as well as DNA replication in this image. This image has a lot of good detail, and I would recommend taking some time to study it. You can see the DNA within the nucleus here, DNA replication here with deoxyribonucleoside triphosphates. Transcription is here, RNA polymerase, as well as the ribonucleoside triphosphates. Then the RNA is processed with the removal of introns. The RNA leaves the nucleus and translation occurs in the cytoplasm. Ribosomal RNA is transcribed in the nucleolus to form single-stranded molecules that become, along with protein, the ribosome. We can see tRNAs and amino acid monomers in the cytoplasm. And over here, again, we can see the formation of the polypeptides. And lastly, let's take a quick look at mutation with a focus on one specific type of mutation, known as a base substitution mutation, that forms sickle cell anemia. Here are the IB syllabus statements. Define gene mutation. Explain the consequences of a base substitution mutation in relation to the process of transcription and translation, using the example of sickle cell anemia. The GAG has mutated to GTG, causing glutamic acid to be replaced by valine, and hence sickle cell anemia. A mutation is a change of the nucleotide sequence of a gene within the genome of an organism. Mutations result from unrepaired damage to DNA in the process of replication. Mutations can be caused by radiation or chemical exposure, but once in place within the genome, they become inherited. And you can see a change in DNA strand 2 as compared to DNA strand 1. Mutations occur during DNA replication and they become heritable if the mutation occurs during replication in a germ-line cell, in other words, a cell undergoing meiosis in the ovaries or testes. In this diagram, we can see the original DNA here, the transcribed mRNA, and the resulting amino acids, including proline. Let's focus to the right side of the image. Here is the mutation in the mutated DNA strand, and here is the transcribed RNA and the resulting amino acid sequence with threonine rather than proline. A single base change can have dramatic effects on the functioning of a protein and one's phenotype, including the possibility of disease. In this diagram, the mRNA molecules are shown. The RNA from the normal gene is shown here, while the RNA from the mutated gene is shown here. The base substitution mutation occurs here. The base in the RNA strand from the normal gene is a G, thus the codon is GGC, coding for the amino acid glycine. But the mutated RNA strand has an A instead of a G. This is a base substitution mutation. The codon is AGC, and the amino acid is serine, not glycine. A single amino acid change, even in a long strand of amino acids, can have dramatic effects on the functioning of the protein to the extent of causing disease. This diagram shows the sickle cell mutation, and we'll start with the amino acid sequences at the bottom. The sixth amino acid in a chain of 146 in normal hemoglobin is glutamic acid. The mutation results in glutamic acid being replaced by valine. One single amino acid change in 146 amino acids results in a disease that has dramatic effects for someone who inherits sickle cell anemia. In this example, the sixth codon in the wild-type DNA is CTC, with the complementary RNA codon, GAG. The GAG codon results in glutamic acid. The mutation changes the second base from a thymine to an adenine. Thus, the RNA codon is GUG, not GAG, and the GUG codon puts valine into position. Now, if you look back at the IB syllabus statement, you might be wondering about the statement as it reads, the GAG mutating to GTG. But you have to think carefully. Mutations involve the sense and anti-sense strands. GAG would be complementary sense strand to this CTC, and the GTG would be the complementary mutated sense strand to this CAC. Now you have to be thoughtful. The IB syllabus is referring to the sense strand. And if you're thinking very carefully, you can see that the sense strand of normal hemoglobin would be GAG, and the sense strand of the sickle cell would be GTG. Maybe the next slide will clarify this for you. So here is the DNA for normal hemoglobin, coding for the second, third, fourth, fifth, and sixth amino acids. Here is the sense strand, and here is the antisense strand. Here is the mRNA transcribed from the antisense strand. The codon GAG brings glutamic acid into position as the sixth amino acid out of 146. Here is the DNA for sickle cell hemoglobin, coding for the second, third, fourth, fifth, and sixth amino acids. Here is the sense strand, and you can see the GTG instead of the GAG in the normal strand. The antisense strand is CAC, the RNA codon is GUG, and valine comes into position as the sixth amino acid. Here is some detail of the sickle cell condition. Normal hemoglobin has glutamic acid in the 6th position, while sickle cell hemoglobin has valine in the 6th amino acid position. This results in a phenotype seen here as a sickled red blood cell. And individuals with sickle cell anemia struggle with exercise and struggle at altitude as oxygen is not well circulated in the body. Remember that hemoglobin has conjugated protein, as the polypeptides are associated with a prosthetic group that includes iron. And that brings us to the end of IB Bio Transcription Translation Part 5.